Let's now have the microarray experiment. Unless you have mastered the basic processes in the central dogma of molecular genetics, this virtual lab might be hard to grasp. It's better that you review it first. But again, I'll try to explain things as we go along in each step of this lab. Alright, so I think it's good that we have some review sections here first before we go into the experiment proper. Let's start at understanding genomics. A genome refers to the totality of the genetic information of an organism. Of course, since the establishment of molecular genetics, most investigators work with individual units called genes. If only one can hold all the genes in his or her hand, then it would be more convenient for researchers to study genes. DNA microarray is one technique that allowed this possibility. It also gave rise to a new discipline called genomics, which is at a different level than genetics, that can only handle one or a few genes that control a particular trait. We know that genes provide the information to make proteins. Every cell in the human body, for example, are identical in terms of the DNA. And the gene sequences, whether it's a skin cell or a muscle cell, or a nucleated baby red blood cell here. But why are these cells different from each other? The concept being presented here is that not all the genes are turned on in all these cells. It depends on what genes are being turned on that gives character to a particular cell. So meaning if we have a cell sample, we only know that a particular gene is active in the cells if there are messenger RNAs present inside the cells. Meaning the genes are being transcribed and used to make proteins. That particular gene is being expressed, so to speak. We have here examples. A muscle cell has the proteins actin and myosin active, while the insulin and the melanin genes are off. We compare this to the beta cells of the pancreas, for example, here. You can see what genes are active in these cells. Using a microarray chip, we can actually detect what genes are active in any given cell as long as we can check for mRNA production or its presence in the cytoplasm. Alright, so it's time to run a microarray experiment to understand the basic mechanisms here. These are the steps to create a microarray. Always keep in mind that a microarray is a chip or a slide that contains all the genes of a particular organism that you sequenced. I will try to explain further how a microarray is prepared. First, you sequence the entire genome of the organism that you are interested in. This means you have to sequence every chromosome. The human genome, for example, has over 3 billion base pairs that can be divided into 23 pairs of chromosomes. This can be different when you have other organisms. A genome is just a sequence of letters, so you need to find the sequences with meaningful information called genes. Now, the goal of knowing all the genes is of course to be able to amplify each gene so you can get adequate amounts to place on your microarray slide. Now, you can only do this via PCR, so you must also create all the primers needed for all these genes. Now, in step 4, you will now be performing PCR and you'll get sufficient amounts of gene fragments that you can stick on your microarray slide. So imagine like doing, let's say, 10,000 PCRs or even more. So this step is easier said than done. Expect plenty of failed attempts in these steps. So step 5 and 6 will help in ensuring that all genes are reproduced and with no defects. So step 5 here, run quality control test on all the gene copies that you make to find out which PCR reactions didn't work. Step 6, you repeat steps 3 to 5 as needed to make copies of all the genes that didn't work the last time around. Let's now go to step 7. Now, if all genes are ready, you must split them into single strands. Only single strands will be placed in the microarray. The microarray must be sticky so that our sample cDNA later can stick to its individual gene spots. and can be detected during analysis. Step 8, when you have prepared the single strands, that's the time when you can plant them on the microarray slides using robots. In this picture, they call it robotic printing. And the remaining steps 9 and 10 must ensure that each spot has comparable quantity of DNA and it can be tracked and managed by a computer for later analysis. In this virtual lab, we already have a human microarray available, so we're lucky. Now, that's a spot in a microarray with your planted single-stranded DNA for every gene you identified in the entire genome. In this experiment, we will compare a cancer cell with a normal cell. In theory, both can look the same, but since cancer cells undergo unregulated cell division, it must have genes that are turned on when these genes are supposed to be turned off in normal cells. Now, once we know which of these genes are turned on, we can easily detect cancer in a tissue sample using a microarray. So let's go and check if our initial assumptions are correct. Here again, both cells look the same. But at the molecular level, we have a gene here that is functioning abnormally. This probably is a protein that regulates cell division. So once it's defective, it can no longer produce a protein that can regulate cell division. Now, these are the 7 steps for this experiment. Let's have all this necessary equipment and reagents ready. Let's collect a cancerous tissue and a healthy tissue. Next is to break the cells to release the RNA and other cell components using solvents. We have this vortex mixer here to agitate our samples uniformly and efficiently. Then of course, like what we did in DNA extraction, we will also centrifuge our samples to separate the RNA from the remaining cell debris that we are not interested. Here we have RNA that's left in the solution. The rest settled at the bottom of the tube. You might ask, why is DNA not also in the solution like what we have last time in DNA extraction? Take note that we did not digest the proteins associated with DNA, so we have sort of a heavy protein DNA complex in the sample. So the DNA settles with the protein at the bottom. Now, we will transfer the RNA solution to another container. We have RNAs here, all the RNAs. But again, we only need the messenger RNAs or mRNAs, right? Now, how do we separate them from the mixture? mRNAs have this unique poly-A sequence. So guys, it's a series of adenine nucleotides at the end of every RNA or messenger RNA. We can perhaps use a molecule to bind to this poly-A tail, so we can fish out the mRNAs from the mixture. So here, we let the solution pass through these beads with poly-T sequences. Recall that adenine binds to thymine, so the beads will hold our mRNAs as the remaining other RNAs will flow down the drain. Very clever idea. We now have extracted the mRNAs for both the cancer and healthy tissue. We can now release them from the beads using a buffer solution. Now it's time to color code our samples with a fluorescent dye, green for healthy, red for cancer. So coming with the dye are labeled nucleotides, reverse transcriptase enzyme, and poly-T primers. In other words, you will convert this RNA into your cDNA or complementary DNA. In this step also, the original RNA will be degraded, so we will be left with the cDNA that is tagged to a specific color code. It's now time to apply this to our microarray slides. So if we have a cDNA in our sample that corresponds to an active gene, it will hybridize or partly bind with a particular spot in the microarray. Since they are labeled, they are visible and can be detected by a scanner attached to a computer. Okay, we can have each gene in each spot, then we can monitor the names of each gene in a computer. Let's now pour both solutions. And this is how we can model the process. Spots that only accepts labeled DNA from healthy cells will be colored green. Those with only cancer genes are red, while those spots that bind both will appear yellow. Now we will wash excess DNAs first before analyzing. And let's place it in a scanner. Okay, there are lots of green spots here, but of course, not all are green. These are the ones that are active in normal cells. Let's now look at the genes that are active in cancer cells. Then we will know the common spots for both. These now look yellow. Of course, we cannot really extract additional information from these genes that are common to both. The non-overlapping spots, however, can surely tell us which genes are activated in cancer cells that are supposedly turned off in normal cells. Red spots are turned on in cancer cells, while green spots are turned off in cancer cells. Let's now apply what we have found out to answer these questions. Of course, here, we will choose red because there's increased activity in these genes in cancer cells. Here we have lots of red, but in most cases, it's because these genes are activated by one or two of these red dots here. It doesn't mean that all of these are really independent of each other, or they are independently active in cancer cells. One gene here called gene 4263 activates all these red dots and also turn off these genes in green dots. But again, in a microarray, when all of these red dots are up, you cannot really tell which one is the problematic gene. On the other hand, we have gene 6219 here that is turned on in normal skin cells, but it was defective in a cancer cell. Of course, it will still look green because it can still produce the mRNA in healthy cells. In cancer cells, even if it's defective, it will still be turned up. So obviously, upon overlapping these two, it will look yellow in the microarray. Again, this is one limitation of the microarray. It only utilizes RNAs in the analysis. As long as RNAs are produced, it will all turn up in the scan. The microarray cannot tell us that it is defective in a cancer cell. If you really want to check for damaged proteins because of the damaged genes or defective genes, you should perform a separate analysis that monitors the protein products. So another technique to cover this limitation is suggested here. And again, you can access them in the University of Utah genetics site. So before we part ways, again, let's consider microarray as a powerful tool to detect what genes are active in different cell samples that we want to compare. In our analysis here, both can produce the mRNA for a particular protein, and we can say that nothing is wrong with that gene 6219. What we do not know is that mRNA could be defective and a dysfunctional protein can be produced. So microarray will never tell us that. We cannot also tell which genes went bad or mutated. Again, we only have an array of genes that are active for a particular cell sample. Microarray doesn't check the proteins, only the genes. And obviously, we cannot cure any disease using this gene activity diagnostic technique. Again, I hope that this was a useful tool. Thank you very much for watching, and try to figure things out yourselves by reperforming this virtual lab. See you soon. Bye!